Hey everyone, and welcome. Have you ever thought about the universe that's happening inside you right now? We're talking about an invisible world that powers every single living thing. And today, we're going on a journey to explore it all, from the fundamental rules of life to the incredible, complex machinery of the cell. So let's dive right in.
All right, so here's our plan. We're going to start with the biggest rule in all of biology. Then we'll look at the two main types of cells, see what makes plant and animal cells different, meet a few cells with some seriously specialized jobs, and finally get into how cells move big stuff around. Okay, first up, life's fundamental rule. So where do you even start with something as huge as life? Well, you start with the foundation, the bedrock of all biology. It's called cell theory. And honestly, don't let the word theory fool you. This is more like the non-negotiable rule book for every living thing out there.
Rule number one is actually super simple, but it's also incredibly profound. Here it is. All living things are made of cells. That's it. From the tiniest little bacterium to a giant redwood tree to you to a blue whale, if it's alive, it's made of cells. Period. No exceptions. It is the universal building block of life. Okay, so what does that actually mean? It means the cell is the smallest possible thing that we can point to and say, yep, that's alive. Think of it as the minimum viable product for life. It's got everything it needs to do the stuff of living, eating, growing, reacting. Anything smaller than a cell, it's just, well, a bunch of chemicals. It's not alive. And the third and final rule is a big one. Cells don't just pop into existence out of nowhere. Every single cell on this planet came from another cell that divided. That means there's an unbroken chain of life stretching all the way back for billions of years to the very first cells. The bottom line is simple. Life comes from life. So we have this amazing, simple theory that unites all of life. But it brings up a really obvious question, right? If everything is made of cells, are all those cells basically the same? And the answer is a huge, resounding no. And that's where things start to get really interesting, because it leads us to the first major split in the world of cells. All right, section 2, the two types of cells. Let's get into that first great divide. Turns out, life on Earth really only uses two basic designs or blueprints. You've got your simple model, the prokaryotic cell, and then you've got the deluxe, much more complex model, the eukaryotic cell. Okay, this chart really breaks it down for us. The biggest difference, right off the bat, is the nucleus. See, prokaryotic cells, think bacteria, are pretty minimalist. Their DNA just kind of hangs out in the main space. But eukaryotic cells, the cells that make up you, me, plants, fungi, we're way more organized. We keep our DNA locked up safe inside a command center, the nucleus. And that's not all. Eukaryotic cells are also packed with all these little specialized rooms called organelles, like mitochondria for power. The analogy is perfect. It's literally the difference between a one-room studio apartment and a huge mansion with a kitchen, bedrooms, a power generator, the whole shebang. All right, let's zoom in again. We're going to stay inside that eukaryotic mansion world for a bit, because even here, there's another big split. This one is all about lifestyle. You've got the ones that stay put and make their own food, plants, and the ones that have to move around and eat, animals. And what you see here is a classic case of form follows function. A cell's design is perfectly suited for its job. Take a look at the plant cell. It needs to be sturdy, right? So it's got this tough, rigid cell wall. It makes its own food, so it's packed with chloroplasts, little solar panels. It needs to store water, so it has a huge vacuole. Now compare that to the animal cell. It needs to move, so no rigid wall. It's flexible. It eats food, so no need for chloroplasts. But it does need a way to break that food down, and that's where lysosomes come in. They're basically the cell's stomach and recycling plant all in one. So, we're not done zooming in. Even inside one single plant or animal, not all cells are created equal. They actually choose a career path. It's a process called differentiation, and become total specialists. So, let's meet a few of these pros. First up, let's hit into the plant world and meet the water seeker, the root hair cell. This guy has one job and one job only: soak up as much water and as many minerals from the soil as it possibly can. So how does it get so good at its job? It's all in the design. It grows this super long, skinny projection, that's the hair part, and that one simple change massively increases its surface area. Think about it. More surface means more contact with the soil, which means it can grab every last molecule of water it can find. It's incredibly efficient. Okay, now let's jump over to the animal kingdom and look at a specialist that, well, you literally can't live without: the red blood cell. This cell is the ultimate delivery driver. Its one mission in life is to pick up oxygen in your lungs and drop it off to every single other cell in your body. And to be the best at what it does, this cell makes a truly incredible sacrifice. As it gets ready for work, it literally throws out its own nucleus, its brain. Why on earth would it do that? To make more room. Every bit of space is packed with hemoglobin, the protein that actually grabs onto oxygen. It's all about maximizing cargo space. But this also means it can't fix itself or make copies. It's a one-way trip that lasts about 120 days. Your body is churning out millions of these things every second to keep up. It's just a wild piece of biological engineering. Okay, for our final act, we are going to the most zoomed-in level yet. We've talked about what cells are, the different kinds, the specialists, but now we're asking, how do they actually move big things in and out? How do they handle security at their border, the cell membrane? Well, when a cell wants to bring in something big from the outside, say a piece of food or maybe even an invading bacterium, it uses a trick called endocytosis. The best way to picture this is to imagine the cell literally reaching out and swallowing it. It's like the cell is eating or drinking things from its environment. And it's not just a clumsy gulp. It's this really elegant step-by-step process. First, the cell has to recognize what it wants to grab. Then, its membrane starts to sink inwards, creating a little pocket around the target. That pocket gets deeper and deeper until pop! It pinches off from the outer membrane, forming a little package, a vesicle, that's now safely inside the cell. And the cell has two main moves here. There's phagocytosis, which literally means cell eating. This is for the big, solid stuff, like when one of your immune cells gobbles up a nasty bacterium. Then there's pinocytosis, or cell drinking. This is when the cell just wants to take a sip of the fluid outside, bringing in whatever little dissolved molecules are floating around in it. But bringing things in is only half the battle, right? Cells are busy places. They also need to ship things out. We're talking about waste or important messages like hormones. And to do that, they basically just run the whole process in reverse. It's called exocytosis. You could think of it as, well, cell spitting, but in a very, very organized way. So imagine a little package, a vesicle, filled with whatever needs to be exported. It travels up to the cell's outer edge, docks with the membrane, like a ship at a port, and then the two membranes just merge. They become one. This opens up a little doorway to the outside, and poof, the contents are released. This is happening constantly. It's exactly how the cells in your pancreas release insulin every time you eat sugar. So there you have it. We went from one single rule for all of life, all the way down to the intricate dance of molecules at the cell's border. And the most mind-blowing part? All of this, the specialized cells giving up their own command centers, the membranes constantly eating and shipping things out, all of it is happening inside you, trillions and trillions of times every single second, without you even thinking about it. It just makes you stop and wonder, right? What else are our cells up to that we don't even know about yet? Have you ever just stopped and thought about how a single, tiny cell knows how to build an entire organism? You know, everything from a microscopic bacterium to a massive blue whale. Well, the secret is locked inside one absolutely incredible molecule, DNA. Today, we're going to unpack this master blueprint of life and really see the elegant machinery that makes it all work. So here's the big question we're tackling. How on earth does this one microscopic double helix pull off the monumental task of not only building a living thing, but also copying itself with near-perfect accuracy, literally trillions of times over? Let's dive in and find out. Okay, first things first. Before we can understand the entire blueprint, we need to know what it's even made of and where all this unbelievably important information is kept. Right, so DNA, that stands for deoxyribonucleic acid, is the master plan. Seriously, think of it like the ultimate instruction manual for life. And it's so precious, it's kept safely locked away inside the cell's command center, its own little vault, the nucleus. From there, it directs every single thing that happens. So what is this master plan actually written on? It seems so complex, but this enormous molecule is built from some surprisingly simple repeating parts. Let's break it down to its most basic components. Everything starts with this one tiny piece called a nucleotide. You can basically think of it as a single Lego brick. And every single one of these bricks has three distinct parts. You've got a phosphate,
a sugar called deoxyribose, and then a nitrogenous base. And it's this base that gives each Lego brick its unique identity.
And here's the crazy part. There are only 4 flavors of these bases in DNA: adenine, guanine, cytosine, and thymine. We just call them A, G, C, and T. It is wild to think that these 4 letters make up the entire alphabet of the genetic code. The specific order of these letters, stretching for millions or even billions of units, spells out every single instruction for building and running a living thing.
Okay, so how do all these individual bricks connect to each other? Well, the sugar of one nucleotide links up to the phosphate of the next one over and over, forming this really strong chain. We call it the sugar-phosphate backbone. This is what creates the long, sturdy sides of the DNA ladder. All right, we've got the sides of our ladder. Now let's see how this whole thing comes together to form DNA's iconic and frankly beautiful structure, the double helix.
Now, this is where it gets really clever. There are strict rules to building this thing. The bases, which are the rungs of our ladder, they don't just pair up randomly. Nope. Adenine always pairs with thymine, and they're held together by 2 hydrogen bonds. And guanine always pairs with cytosine, using 3 hydrogen bonds. These are called Chargaff's rules, and they are absolutely non-negotiable. It's this strict pairing that keeps the code consistent. And here's another key design feature. The 2 strands of the DNA ladder are anti-parallel. Just picture a highway with 2 lanes of traffic going in opposite directions. It's the same idea here. One strand runs in what we call the 5' to 3' direction, and the other runs 3' to 5'. All you need to remember is that they run in opposite directions. This detail might seem small, but trust me, it is absolutely critical for how DNA gets copied.
So let's put it all together in what we call the Watson-Crick model. You've got 2 strands coiling into that famous double helix. The strong sugar-phosphate backbones are on the outside, kind of like a protective railing, and all those important AT and GC base pairs are tucked safely on the inside. This twist also creates these little grooves that let other proteins get in and read the genetic code. Okay, so a static blueprint is one thing, but the real magic of DNA is its ability to make a perfect copy of itself. Let's shift gears from the structure to the action and see the incredible, dynamic process of replication. The core principle here is that replication is semi-conservative. It's a fancy term, but the idea is simple and brilliant. The cell doesn't build a new DNA molecule from scratch. Instead, it unwinds the original and uses each of the 2 old strands as a perfect template to build a brand new partner strand. So every new DNA molecule is half old, half new. It's just an incredibly efficient and accurate system. Now, this process doesn't happen on its own. It requires a whole team of tiny molecular machines we call enzymes. Let's meet the crew. First up, you've got helicase, the unzipper. Its job is to split the 2 strands apart. Then you have primase, the initiator, which lays down a little starting block. The star of the show is DNA polymerase, the builder. This one adds all the new nucleotides, and get this, it even proofreads its own work. And finally, there's DNA ligase, the gluer, which we'll see is crucial for finishing the job neatly. The whole show basically follows a simple 3-act structure. It kicks off with initiation, where all the machinery gets assembled at the starting line. Then comes elongation. That's the main event, where all the new DNA is actually built. And finally, it all wraps up with termination. Okay, now for the trickiest part. Remember that whole anti-parallel thing, the 2 strands running in opposite directions? This is where it really matters. See, the main builder, DNA polymerase, can only work in one direction. So for one strand, the leading strand, it's smooth sailing. It just builds continuously. But the other strand, the lagging strand, runs the wrong way. So the polymerase has to work backwards, building it in short, choppy pieces called Okazaki fragments. It's kind of like trying to paint a stripe on the road while you're driving in reverse. You have to do it in little bursts. And this is where our final enzyme, DNA ligase, becomes the hero. After the polymerase builds all those little Okazaki fragments on the lagging strand, DNA ligase comes in and acts like molecular glue. It seals all the little gaps in the backbone, turning that choppy, fragmented strand into one single perfect piece of DNA. Job done. So, we've just walked through this incredible molecular dance. It's pretty amazing, right? But why does it matter? Let's zoom out from all the enzymes and nucleotides and see the profound impact this one process has on, well, all of life. Think about it. Every single time you heal from a cut
or grow just a little bit taller, or simply live from one day to the next. It's all because of DNA replication. It's the fundamental process that ensures when one of your cells divides, both of the new cells get a perfect copy of the genetic instruction manual. It's essential for growth, for repair, for reproduction, and for passing your traits on to the next generation. And here's a fascinating little twist in the story. The process is almost perfect, but not quite. Once in a while, errors or mutations can happen. And while that might sound bad, this quote reminds us of something incredible. These very mistakes are the raw material for evolution. They create the genetic variety that allows life to adapt and change over millions of years. So just let that sink in. This unbelievably complex, yet nearly perfect copying process is happening inside you right now, billions and billions of times over. It is the engine of life itself. But that leaves us with one final, crucial question to ponder. Given the sheer scale of it all, what are the real consequences when this masterful process inevitably makes a mistake?
Welcome to The Explainer. You know, we're all built from a set of instructions, a kind of biological blueprint. Today, we're going to decode that instruction manual. We're going to look at the incredible, microscopic process that takes the genetic code, the stuff locked inside your DNA, and turns it into, well, you.
So here's our game plan. We're going to follow the journey of a gene all the way from the master blueprint in the DNA to the final protein that actually does all the work. We'll look at the code, how we make a messenger, the two big steps, transcription and translation, and finally, why proteins are, well, everything.
So how does a single cell even know what to do? How does it build an entire person? Well, it really boils down to a huge logistics problem. Think about it like this. Imagine you've got the master plans for a skyscraper. They're priceless, so they're locked away in a super secure vault. But the actual construction site, with all the workers and the steel and the concrete, is miles away. So how do you get the instructions from the vault to the builders without risking the original blueprint? Your cells face this exact same problem every single second.
Okay, so let's start at the beginning. We're diving into the cell's central command center. You know, the vault where that master plan is kept?
Right, so here's the deal. Your DNA, that's the master blueprint for everything about you, is locked up safe and sound inside the cell's nucleus. It can't leave. It's too important. But the factories that actually build things, the little protein-making machines called ribosomes, they're way out in the cytoplasm, the main part of the cell. So the DNA is stuck in one room, and the factories are in another. We've got to find a way to get the instructions across that gap.
And the cell has this absolutely brilliant solution. It never ever lets the original blueprint leave the vault. Nope. Instead, it makes a temporary, disposable copy of just the one page it needs for the job. And this little messenger molecule is called RNA. RNA is the hero of our story today. It's the key to this whole operation.
Okay, now this is where it gets really cool. Look at how perfectly RNA is built for its job as a temporary messenger. DNA, you see, is double-stranded and super stable. It has to be. It's the long-term storage. But RNA, it's usually single-stranded and much less stable. And that instability is actually a feature, not a bug. It means the message doesn't stick around forever, so the cell can control exactly how much of a protein it makes. And check this out. Instead of the base T, thymine, which is in DNA, RNA uses U for uracil. It's like a chemical stamp that says, hey, I'm just a temporary copy. Don't mistake me for the real deal.
And get this, it's not just one type of RNA. It's a whole specialized team, a whole crew. You've got mRNA, messenger RNA. That's our blueprint copy. Then you have tRNA, transfer RNA. Think of these as the delivery trucks, each one carrying a specific building material. And finally, there's rRNA, or ribosomal RNA, which is actually part of the factory itself. It's what the ribosome is made of.
So you might be wondering, how does the cell actually make that messenger copy? Well, this first big step is a process called transcription. Just imagine a scribe carefully entering that vault to copy one single page from the master blueprint. That's transcription.
So a special enzyme, it's called RNA polymerase, acts like our scribe. It finds the exact spot on the DNA where the gene begins. Then it carefully unwinds a little section of that famous double helix. As it moves along one of the DNA strands, it builds a brand new matching RNA strand, letter by letter. It's an almost perfect copy. And then, once it reaches a stop signal on the DNA, it just lets go, releasing our freshly made mRNA message.
But wait, it's not quite ready for the outside world yet. Before that mRNA copy leaves the safety of the nucleus, it gets some editing.
First, it gets a protective cap on the front and a long tail on the back. This helps it survive the journey and tells the factory where to start reading. Then, something amazing happens. The cell snips out all these non-coding bits called introns. The parts that are left, the good stuff, are called exons. This splicing process is incredible because it means one gene can be edited in different ways to create instructions for multiple different proteins. It adds a whole new layer of complexity. Once that's done, our message is lean, clean, and ready to go.
All right, our messenger RNA is on its way. But before we get to the factory floor, we have to talk about the language. Because the cell doesn't read this message one letter at a time. It reads it in words.
Yeah, the entire genetic language is based on three-letter words. We call them codons. Every single group of three letters on that mRNA, like say AUG or CCA, is a code for one specific amino acid. And amino acids, remember, are the building blocks of proteins. So the codon AUG, for example, is basically the universal signal for start here, and it also calls for the first amino acid, which is called methionine.
Okay, so if the mRNA has the word, how does the right building block show up? This is where our delivery trucks, the tRNA molecules, come back into play. Each tRNA carries one specific amino acid on one end, and on the other end, it has a three-letter sequence called an anticodon. This anticodon is the perfect complementary match for a codon on the mRNA. It's like having a key that only fits one specific lock. It's how the delivery truck knows exactly which spot on the assembly line to drop off its cargo.
Okay, this is it. The big moment. Our edited mRNA message is out of the nucleus, it's found a ribosome, one of those protein factories. We understand the language. It is time for the second major step, translation. This is where we literally translate the language of genes into the language of proteins.
This is the cellular assembly line in all its glory. The ribosome clamps onto the mRNA and finds that start codon. The first tRNA delivery truck pulls in and drops off its amino acid. Then, the whole ribosome shifts down the line by exactly 3 letters, one codon. The next tRNA, the one with the matching anticodon, pulls in. The ribosome then forges a powerful chemical bond between the 2 amino acids. This is called a peptide bond, and it's the stitch that holds the whole protein together. The ribosome just keeps chugging along the mRNA, reading a codon, bringing in a tRNA, linking the amino acids over and over and over, until it hits a stop codon. Then, the whole thing comes apart, releasing a brand new protein chain.
So let's recap. We went from a gene in the DNA to a messenger RNA copy, and now we have this long chain of amino acids, which we call a polypeptide. That's a ton of work for one tiny little chain. So why? Why does any of this matter? Because that chain isn't the final product. It's about to fold itself into a protein.
See, that simple chain of amino acids is about to do something magical. It folds up into a very specific, incredibly complex 3D shape. It's like molecular origami. And that shape is what makes it a protein. And what do proteins do? It's almost easier to ask what they don't do. They are the enzymes that speed up every chemical reaction in your body. They're the structural components, like collagen. They're the transport crew, like hemoglobin carrying oxygen. They're the messengers, the defenders, the movers. They are literally the machinery of life.
And this whole thing, this elegant dance of transcription and translation, is called the central dogma of biology. DNA makes RNA and RNA makes protein. It's happening in trillions of your cells right this very second as you're listening to this. It's the fundamental process that takes the static code of your genes and turns it into the dynamic, living, breathing you.
And look, this isn't just some abstract science lesson. Understanding this process down to the letter is changing our world. Think about it. We've learned how to design our own mRNA messages, and that's how we got the revolutionary COVID-19 vaccines. We're figuring out how to fix mistakes in this process to treat genetic diseases. We've decoded the instruction manual of life, and now we're starting to learn how to write the next chapter ourselves.